The paradox of viruses. What are they made of? While viruses may not be considered alive, they share many biological similarities with living cells. They are also made of the same building blocks. But what are they made of? Viruses are made of DNA or RNA protected inside a protein shell. Viroids don't even have the shell. DNA is made of nucleic acid molecules strung together in a double helix chain. And molecules are made of atoms. But what are atoms made of? And how do they connect together to form molecules? Electrons are a fundamental component of matter. They are also the international unit of measurement for electric charge. Electrons basically are electricity. They have an equal and opposite electric charge to protons, although neither is inherently positive or negative. Electrons are attracted around the protons and arrange themselves more like an electromagnetic field of layers around a magnet than solid planets orbiting a sun. This forms atoms, and the number of protons in the center determines which element on the periodic table it is. Each element can only hold so many electrons worth of energy in each layer of its electromagnetic field. Each layer further out from the nucleus can hold more energy. When the outer layer is full, the atom is not chemically reactive. It is balanced, self-sufficient, and stable. Examples include the so-called inert gases, like helium and neon. They are so unreactive that they would not join together to form liquids or solids. While helium is the second most abundant atom in the universe, we are running out of it here on Earth. Only several helium atoms are found per million atoms in our atmosphere. That's because it does not connect with other atoms, it just floats off. Neon has 8 electrons in its outer layer and can fit no more, making it chemically stable. For some unknown reason, if any atom has 8 electrons worth of energy in its outermost electron shell, it behaves as if it were full, even if it could fit many more. Not only that, but atoms are willing to share or even give away electrons if it will bring their outer layer to the magical number of 8. Reminiscent of the octave in Western music, this odd characteristic of matter is known as the law of the octet and is the basis of the periodic table. The significance of this electromagnetic bonding between atoms cannot be overstated. It gives rise to matter itself. While gravity pulls you down, the electromagnetic bond between atoms in the ground push you back up. Over a short distance, the electromagnetic force is stronger than the gravity of our entire galaxy. Molecules. Biological systems are made of matter, that is, atoms made in the Big Bang or stars. The 4 most abundant yet reactive atoms in the universe are also the 4 most common in living things. What a surprisingly obvious fact. In your body, for example, about 65% of the mass is oxygen, 18% carbon, 10% hydrogen, and 3% nitrogen. Excluding water, the numbers for an average cell jump up to almost 50% carbon, 30% oxygen, 9% hydrogen, and 8% nitrogen. Viruses are made of these same building blocks. Carbon atoms are very good at making connections. Indeed, there are more carbon-based molecules than all other known molecules combined. All biological systems of Earth are carbon-based, including viruses. One example of a carbon-based molecule is nucleic acids, the building blocks of DNA, which are actually several molecules attached together. The center of the molecule is a pentagram ring of 5 carbon atoms, forming a sugar known as ribose by linking with hydrogen and oxygen atoms. Another molecule forms from a phosphorus atom surrounded by 4 oxygen atoms. These 2 molecules are linked together by sharing electrons and alternate to form long chains. This is the structural support or scaffolding for the genetic code. While the sugar molecules can come in both varieties, in all cells and viruses on Earth, they twist exclusively to the right. This is known as chirality and causes the chains to form spirals. Attached to the side of these chains is another molecule made of 1 or 2 rings of nitrogen, carbon, hydrogen, and some oxygen. They come in only several varieties, and each type will only connect with one other. That's because the atoms are seeking to fill their outer electron shell, and, like a lock and key, their structure gives them only one possible way to lock together. These pairs are attracted together magnetically. On the most fundamental level, our genetic code, like computer code, is binary. These pairs then attach 2 spiral chains together, like the rungs of the now famous double helix, or attached to another part of the same single chain. This is DNA and RNA respectively. The molecular subunits of DNA have spontaneously emerged in labs by heating and zapping the right mix of atoms in the right way. They've been found in the 7 billion year old Murchison meteorite, which is older than our sun, and maybe even in water geysers spraying off of one of Saturn's moons. So while Earth may be the only home to life in our galaxy, it is not the only home to the building blocks of life. Genetic code. Most scientists think of life as a chemical process, but could it also be described as a gathering of information? While based on the past, the information must be useful for future survival. Our genetic library is a collection of information about the world we live in and how to survive it, gathered slowly, bit by bit, over billions of years of evolution. But before we could collect information about how to survive in a complex world, life first needed an alphabet to write in. The letters of the alphabet are the nitrogen bases in nucleic acids. These link together to form words, then sentences, and then different stories about what life could be like. Any chain of nucleic acids has only 4 different letters: A, U/T, C, or G. Combining letters makes a genetic word called a codon, which is the instructions to build a specific amino acid, the building blocks of proteins. 1-letter word would only give 4 choices. 2 letters would give 16, and 3 letters would give 4 times 4 times 4, which is equal to 64. This is more than enough for the 20 amino acids essential for life. So there are only 20 words and 64 different ways to spell them. For example, CGU, CGC, CGA, and CGG all mean the amino acid arginine. Not exactly perfect penmanship, but it gets the job done. Multiple words are then strung together to form sentences, called genes, which are usually the instructions to build a specific protein made of multiple amino acids. A single gene can be hundreds to millions of codons long. Not exactly easy to follow, yet the same genetic language is understood by all known living things, even viruses. As many sentences are added, a story begins to form. A chapter is called a chromosome, and the entire book is called the genome. The shortest books are the instructions for viruses, and the shortest has only 4 sentences about making 4 proteins. Even the influenza virus, responsible for the common flu, has only 14 protein coding genes. Is this still a book, or merely a pamphlet? The human genome, on the other hand, was first read in the year 2000 and was found to contain about 46 chromosomes, 25,000 genes, 1 billion codons, 3 billion nucleic acids, and many billions of atoms. If you typed out the story and printed it off, the stack of paper would be 100 meters high, or as tall as a 30-story building. Because the genetic letters are usually enclosed within 2 strands, DNA is a very stable way to store information. Indeed, copying information by DNA is more accurate than any current computer. When not in use, DNA winds up tightly to protect the information even further, making it a very compact way to store data. If unwound, the DNA in each of your cells would be taller than you. In recent years, binary computer code can now be converted into DNA. A few months ago, all of the words from all of the Wikipedia articles in English were stored on a tiny amount of synthesized DNA. If converted to DNA, the whole internet could be stored in a filing cabinet. However, the speed of access would be slower than dial-up internet and more expensive than an F-35 jet fighter. We humans are remarkable creatures with adaptable brains and complex immune systems. We have 8 times more genetic code than a type of pufferfish, 5,000 times more than the simplest bacteria, 100,000 times more than the coronavirus, and 500,000 times more than the simplest virus. Yet the genome of onions or wheat is about 5 times longer than our own, and the printed version would be taller than the Empire State Building. The broad-footed salamander genome is 20 times longer than our own, and a small flowering plant in Japan is 50 times longer. A single-celled amoeba called Polychaos dubium is 200 times longer. But bigger is not always better, and more words do not always make a better story. So I better stop waffling and get to the point. The genome size is not proportional to the complexity of the living creature. This became known as the C-value paradox. The paradox was resolved by the discovery that most of our genome does nothing. Only about 1% of our genome directly codes for making proteins, and the difference in genome size is mostly proportional to genome efficiency. For example, one species of plant can fit about the same number of genes as us into a genome 36 times smaller than our own. That's because 97% of the genome codes for proteins, compared to just over 1% for us. This efficient little carnivorous plant, known as a floating bladderwort, is able to delete unnecessary parts of its genetic code and now thrives in all continents except Antarctica. Pelagibacter ubique, perhaps the most abundant bacteria on the planet, is also one of the smallest and simplest self-sufficient cells. This streamlined sea creature has almost no duplicate genes or otherwise useless DNA. It can make all 20 of the biological amino acids with a genome thousands of times smaller than our own. Humans, on the other hand, can only make about half of them. All viruses have very small and very efficient genomes, more so than all but the most economical of cells. That's because they carry less baggage and commandeer help for the more complex tasks. Which is better? Which are better instructions? Scientists once thought that non-coding DNA was junk that had no purpose, but further research showed that coding for proteins was not the only purpose of DNA. DNA is also responsible for running the protein-making factory. This includes a control center and scaffolding for the processes. Based on biological activity, scientists now estimate that 8 to 25% of our genome is useful. Just because we don't know what it does yet doesn't mean that it doesn't do anything. But even 25% is not very efficient. 3/4 is just useless baggage. Like harmless parasites or male nipples, they are just along for the ride. Indeed, about 8% of our genome literally comes from viruses who infected the reproductive cells of our ancestors. So our genome is almost as much viral as it is functional. What does that say about us? Useful genes are like islands of order in a chaotic sea of nucleic acids. Less a design and more a mosaic. Our genome is not perfect, and neither are we. Structure. So what then is the structure of viruses? Viruses are kind of like the nucleus of a cell minus the cell. They are a bunch of nucleic acids connected in long chains, then wound up and wrapped in a protective case. Without lugging around all that machinery to extract energy or make things, viruses are tiny, like a millionth to a billionth of a meter across. Viruses are about 1,000 times smaller than bacteria, which are about 10 times smaller than animal cells. But the largest viruses are bigger than the smallest cells. The nucleic acid chains can be arranged in different structures, and this forms the basis of the Baltimore virus classification system. Type 1 uses double-stranded DNA, like our genome. Examples include giant viruses, herpes, chickenpox, and smallpox, which used to be one of the biggest killers of humans. Type 2 uses single-stranded DNA that must be converted to double-stranded DNA before it can function. Examples include the parvovirus, a common infection in dogs. Type 3 uses double-stranded RNA that separates into single strands before it can function. This includes rotavirus, the most common cause of diarrhea in children and babies. Type 4 uses single-stranded RNA which can directly function as instructions. Examples include coronavirus, i.e. COVID-19, SARS, which is another type of coronavirus, the common cold, called rhinovirus, hepatitis C, yellow fever, Zika, and polio. Of these, coronaviruses have the largest genome. Type 5 uses single-stranded RNA that must be converted to its opposite base pairs before it can function. Examples include influenza, aka the common flu, Ebola, measles, mumps, and rabies. Type 6 uses single-stranded RNA that must be converted to DNA and then inserted into the cell's genome before it can function. Examples include HIV and other retroviruses. Finally, type 7 uses double-stranded DNA that must be converted to single-stranded RNA before it can function. Examples include hepatitis B. In all living cells on Earth, RNA makes a temporary copy of permanent DNA and carries their instructions to wherever they are needed. If DNA is like the long-term storage or hard drive, then RNA is like the working memory or RAM. Yet viruses can use either. and can even reverse the process. So let's take a look at the differences. RNA is a ribose nucleic acid and usually forms single-stranded chains. DNA is deoxygenated ribose nucleic acid, which has one less oxygen atom and one of the nitrogen bases is slightly different, but is otherwise identical. For some reason, these differences make DNA tend to form a double helix. Double-stranded DNA is the most stable, since it encloses the fragile nitrogen bases, and single-stranded RNA is the least stable. This makes RNA viruses more prone to mutation, which is both a blessing and a curse. It will accelerate evolution, but can also lead to error catastrophe, when excessive mutations result in extinction. While the extinction rate of cell species to date is over 99%, the extinction rate of RNA viruses is probably much higher. Hopefully, the coronavirus will mutate itself out of existence sooner rather than later, but it will happen eventually. However, this vulnerability comes with a unique advantage. While most viruses have to convert their genetic code into a usable form, often in a multi-stage process, the RNA of coronavirus can directly tell your cells what to do. This makes coronavirus and other type 4 viruses like the common cold, Zika, and polio highly infectious. It also makes it easier for different strains infecting the same cell to produce genetic shift by shuffling their genes together. This is probably how the novel coronavirus appeared. Perhaps the different strains came from different species like bats and pangolins, then merged together. Protecting the genetic material of viruses, whether RNA or DNA, is a protein case called a capsid. Several identical Lego-like building blocks snap into place, usually forming helixes or icosahedrons. The platonic solids are the only 3D geometric shapes where all faces are the same shape, all edges are the same length, and all meet at the same angle. The icosahedron is the most spherical of the platonic solids and has the lowest surface area to volume ratio. It is made of 20 identical triangles, the simplest 2D shape. This makes it literally the most efficient shape to enclose space made of identical building blocks. This saves space in the viral genome. The coronavirus shell is a spherical helix, while the common cold, amongst many others, is icosahedral. However, most viruses are too small to actually see. Instead, their structure is determined by seeing how it interacts with electricity. Coronavirus has only ever been seen through an electron microscope. Some, but not all viruses, are then enveloped in an oily membrane. This further protects the genetic material encased within, since oil is water resistant. In water molecules, the small hydrogens and big oxygen are so uneven that they create a polar magnet with a positive and a negative end. Oil molecules, however, have hydrogen atoms more evenly distributed around a central carbon atom and do not have a positive and negative end. So water is magnetically attracted to water and repelled by oil. That's why the two don't mix. The virus membrane is usually a two-sided wall, one to keep water out and one to keep water in, and is usually stolen from within the cells they infect. Herein lies the power of soap. It acts as an intermediary and makes water and oil mix. One end of the molecule is attracted to water and the other to oil. Soap molecules flowing by will stick onto the oily virus membrane and tear it apart. As you have probably guessed, coronavirus is one of the rare ones that does have an oily membrane. Washing it with soap can kill it. That is, if you considered it to be alive in the first place.